So welcome everybody. So this is actually now our 5th webinar in this series. Be really cool to see how many people have actually managed to make it to all 5 of them so far. We get very good audiences for these, obviously, but a lot of interest for DOE out there. So in our first webinar, that was from me, and that was basically just talking about why you might want to use DOE and why it's so important to use DOE if you're working with biology. And then James Arpino followed that up with what you could actually use it for, covering some of the more typical uses for DOE, I guess, as opposed to this webinar today, when we're going to be looking at how it can really be stretched into other areas of biological optimization. And then Steve then gave us a talk about how we'd actually get started on things. Before then, in our 4th webinar, we started to get into the depths of what it takes to design and analyze DOEs effectively. So for this webinar, we're changing tacks ever so slightly and looking to see how DOE can be applied to the optimization of genetic code. So this is a space which, frankly, I would never have thought would have been particularly tractable to design of experiments. Design of experiments is usually used for quite smooth design spaces, which have fairly clear ways that you can navigate your way through, and genetic design doesn't necessarily fall into that space. And so I've always found it fascinating to talk with our speaker today about these challenges of optimizing genetic code, and that's why I'm really excited about our webinar today. So I'm really excited to introduce our presenter for today's webinar. He's someone I've known now for quite a long time. We're fellow enthusiasts about multifactorial optimization and DOE approaches. I've known him probably for about 10 years, but actually, he's got over 25 years of experience of engineering biology across academia and industry, and he co-founded Atum almost 20 years ago now. And that's where they've been building out for a very long time these methods for navigating genetic space in a systematic and highly effective way. He himself has extensive knowledge in gene design, protein expression, protein engineering, machine learning, and synthetic biology. And I'm really excited to see his talk today and the particular case studies that he's chosen to showcase from the huge back catalogue that they have at Atum. So thank you very much for joining us, Claes. And without further ado, I'll hand it over to you.
Okay. So let's see that. Thank you very much for that introduction. And let me pull up my screen here and let's see if we can. So let me know if you see the screen. All good?
Yep, we're all good.
Okay, so thank you very much for that introduction. Really appreciate it. I'll talk a little bit about what we've been building over the last few years and how we see that's being propagated and across the world of biology. And I think there are a lot of nice opportunities. And to your point, Markus, it's very cool and a little bit scary to talk to people who are really smart about this, because they people like you tend to poke hole into our logic. So it's always a good challenge. So just a few words about Atum. So we used to be called DNA 2.0 back in the days. We changed name back in 2016. We're based here in beautiful San Francisco Bay Area. We really build synthetic genes, we build proteins, we build cell lines using the DOE, machine learning, and synthetic biology to navigate and build better processes for customers. So everything we do is fee for service. We don't really have any products ourselves, but we have a lot of customers coming into us. I'll do a few case studies, but of course, all these case studies with the customers we do, almost everything of that I can't really talk about, because at the end of the day, the data is owned by the customer. I'll talk about a few examples where we have disclosed things in the public domain, and I think it's kind of explore how we can build these different things. Company was founded back in 2003, and there are 4 founders. I'm one of the co-founders. We all came out of a company called Maxigen back in the days. Today, the company is 150, 60 employees strong. We are based, you see the building here, we're building number 1. We have a second one as well. So it's 100% solar. We're here in beautiful sunny California. So under the warm Californian sun, that drives all the energy. So we're completely solar panel driven. And of course, being in California, we're also set this whole building up so we're very water efficient. Very science driven, have around 40 patents, and we acquired a company called MIGS now a few years ago as well, and incorporated that into the business to really drive towards therapeutic proteins as a fee for service as well. So let's take a step back and look at biology. To your point, Markus, biology is really complicated, and historically, I think most biologists are really trained in sort of deductive science. You're trying to understand mechanistically what's going on. You have a Krebs cycle, you have boxes and arrows, and this carbon goes to that carbon, goes to this carbon, or you're looking at, I don't know, mice or something, and that if the kidney does this and this and that. So all very mechanistically driven. I think what's happened over the last 20, 30 years now is really becoming much more complex and much more complicated, in the sense that the data is so much intensive that we have genomics, we have proteomics, we have metabolomics, and this is for every little cell in the crop in the body. And we certainly have these different spatial and temporal distribution of all these metabolomic and all the ATPs and all the different genes and RNA polymerases and everything. So on that end, it's just this massive ocean of data that didn't used to exist, and I think that it's counter to the biology tradition to look at all this data in a context, because we're really trained as a mechanistic, as deductive reasoning science. At the same time, we also see biology, and certainly biotechnology, is really coming to the intersection between physics, computer science, ecology, chemistry, and so on and so forth, as you're trying to navigate all these different spaces. So what used to be really a deductive knowledge is increasingly becoming inductive knowledge, where you look at, instead of looking at mechanistically understanding how all these arrows and boxes and everything fits together, we get into the point we're looking at where computer science is. Like if you're Google, you don't really try to understand why certain people click on a ad versus other people don't. It's just pattern. It's just pattern recognition and trying to understand what is the correlation between all these different data points. So famously, Norbert Weiner at MIT really talking about the complexity of modeling, and at the end of the day, a model can only be truly reflecting the same entity that you're trying to map out. So the best material model of a cat is another, or preferably the same cat, right? It's really hard to make models that predict complex biological behavior. But having all this data, on the other hand, you can now infer a lot of correlation, causal or not, that affect exactly how you're distributing your data. So to look at that in the context of biology, and I'll get to the whole DOE thing, and I know Markus is waiting. At the end of the day, biology is ACGT, right? That's all there is to it. All the information is captured as ACGT. And we as humans, we think of this ACGT, we build little models, we have little cartoons of how biology works, and we call it a gene, we call it a protein, we call it vector or metabolism or all these different things. But at the end of the day, we all really know that all these models are just simplified stories of what really is going on. Because at the end of the day, even in the Krebs cycle, you have equilibriums and spatial temporal distribution, and things change all. I mean, even this central dogma, DNA goes to protein, goes to RNA, goes to protein. We all know there's things like reverse transcriptase. We all know that all these dogmas are just inferior models. But we can take all this information that's now sitting in the ACGT and build models of how the correlation and the causality of those sequences affect certain outputs. So What I'll talk about is really how we do that on a gene level, on a protein level, on a vector level, and cell level. And that's, of course, how you can design things solely within the DNA. And of course, outside of that box of DNA, then we walk into Synthace land, where we look at, media optimization or shaking speed, or this Erlenmeyer versus that Erlenmeyer. And at the end of the day, how do all these different parameters correlate in order to create an improved product or an improved process, or whatever you are trying to accommodate. So how does biospace differ from sort of the regular Synthace space, or sort of normal design experiment? Well, I think there are three major underlying differences. One being that bio is completely qualitative and extremely defined, right? So whenever we try to do an optimization, there are really two buckets. One is variable selection. How do you know which variables to play with? And the other one is the search optimization, right? So and that's separate. But in the variable selection, in biology, if you look at DNA, you only have 4 variables, A, C, G, T, and if you want to count a void of a variable, so that's the 5th one, right? So that's the complete space, and it's absolute in the sense that you can never have 1.5 adenine. You're always, it's either A or a T. There's nothing in between. The same goes for proteins, right? So you have a total of 20 amino acids, and there's never going to be something that's half valent, half leucine. It's one or the other. That's the only way it is. And of course, you can have a 21st amino acid being a void of an amino acid. We can talk about selenoprotein and all these different things as well. But for the sake of argument here, it's a very qualitative space. And that means that when you're doing the designs, every single virtual point in your DOE space is an absolute defined entity. And same thing if you're actually making this sequence, making this gene, making this protein, it's an absolute sequence. So that makes for some interesting math, and we may get into that at some point, of how you search that space, because everything is qualitative. The other big difference is that in principle, every single experiment has been done by Mother Nature. Mother Nature's put in every single amino acid substitution in every single protein across the last 4 billion years and tried to see if it changed some parameter. And the absolute majority of those experiments failed miserably, and no sequence was able to be generated for that particular protein or that particular gene, because it didn't fold or whatever. So the few sequences that exist today in GenBank are the ones that have survived all these experiments that Mother Nature's been doing. And we can go back and we can pull out all those experiments, and GenBank is just amazing. Here's this fantastic candy box of just all that information you can ever imagine. And it's freely available. It's just best use of my taxes. I smile when I pay my taxes every time, thinking about how cool this is. We get access to this for free. It's just cool. And that allows us then to really use that information in order to see what variables can we play with and which one can we not. Which one clearly is going to blow up the protein, and whereas the ones that do have fruitful functionality, all going to be able to pull that out of GenBank or similar public domain databases. And lastly, I think interesting is epistatic. So what is the synergy between different, say, amino acids in a protein, for instance? Are these two substitutions synergistic or antagonistic, right? And we all know that when we do start playing with biology, we can make a really good substitution in position 1, and we make a really good substitution in position 2, and put the two together, and it just crashes and burns, or vice versa. And of course, this epistatic effects is nicely quantifiable, and there's been a lot of work done on what they call NK landscape. This is sort of Stuart Kaufman stuff from now quite a few years ago, where you can look at what's the synergistic relationship in this epistatic space. And if we assume that K, so the where N is the number of units, so amino acid substitutions in the protein, for instance, and K is the linkage between units. So if every single amino acid talks to every other single amino acid, K equals to N minus 1. But if every single amino acid is linearly additive, K equals to 0. So in a world where K equals to 0, protein engineering would be trivial, because all you have to do is just make all amino acid substitutions, pick the best one separately for each residue, and then just combine them, and you will have the perfect space. So that's sort of the Mount Fuji of biology space. And of course, the other extreme is if everything talks to everything else. There's no way to navigate that space, because the only way to navigate that space is to go and make every single sequence, every single combinatorial sequence of that say protein, and look at the activity. And of course, it's just not physically possible, not even, I mean, barely theoretical possible. So, but you can start looking at the tools, and I'll get into that a little bit, and capture what is the epistatic effect in your typical data set, and then use that to navigate for efficiency. Okay, so just a big picture how we approach this and how we build these things. So we in silico we do design the biological space using typically the DOE, and this is all done in silico. For say we make a protein, we'll define this separately. Then we having access to gene synthesis, we just make typically sets of 96 say proteins, synthesize them, and then we physically, if it's an antibody, we're trying to make a better antibody, for instance, we can just make those antibodies, put them into CHO cells or HEX cells, depending on what systems you have, purify the antibody, protein A columns or whatever, and then take them and do a cell-based assay or many for looking at how well do they bind the target, do they shrink the tumor, do they express well, do they fold, all these different things. And then use the machine learning to capture the correlation between substitutions and causality in order to build prediction models using classic learning. So I'm going to start with a very old and I think kind of cool example. This is really where I started looking at this, and where my brain first popped many years ago. So this is actually a paper that was published in '93. So this originated, and as far as I know, this is the first example of someone using, well, I'm sure Markus will call me heretic for calling it DOE, but and offer my soul to the gods of DOE. But at least it was an attempt to go down that path. And then this being in '93, it was a lot of the tools that we have today were not that easily available. And this started with a friend of mine, Jörgen Jonsson, still back in Sweden. He was a grad student, and I was a grad student in molecular biology. And we were talking about E. coli promoters at the time. And just to show how naive and stupid I was, I made the assumption, and I was fairly adamant about it, that the consensus promoter was the strongest promoter. And he, of course, told me that I was an idiot, and we argued back and forth. Turns out he was right. So we ended up doing this experiment where we identified some previous publications on people looking at different E. coli promoters for T7. So we took those, and with quite a lot of effort back in those days, '93, synthesized those promoters, put them in a vector, and then measured activity as a function of how improved they were over a beta-lactamase promoter sitting on the same vector, and then measure that in coli. And this paper's been published a long time ago. All the data is available. So what you see here, if you kind of squint, you see this as sort of let's call it the pseudo DOE. And I say that because you see these chunks of sequence elements that are represented across this data set, and you see them vary across. But this is really more opportunistic DOE in the sense that how these different promoters were cloned and sequenced and expressed at the time. And we made those, and we measured activity, and some were good, some were bad. And based off of that model, what you see here in the bottom right, we had measured activity versus predicted activity using classic machine learning. This was done using something called PLS, partial least square regression, which is kind of like a PCA with a additional Y dimension. And off of that sequence, we then made 2 additional promoters that we synthesized and tested. And lo and behold, both of them were much better than the training set, validating the model. And of course, none of them were even closely similar to what the consensus promoter was. So seeing this, I realized that, you know, everything I ever learned in biology was a waste of time, and looking at life through DOE sunglasses made everything just, all of a sudden you see the light and you understand how all these bits and pieces fit together, and how life is beautiful all the time. So when we set up Atum back in the days, or DNA 2.0, we started doing synthetic genes, and we did codon optimized sequences. And back in the days, everyone assumed that using the most common codons would be the preferred genes to make. So we had all these people call me up and say, hey, Claes, can you make me this human kinase and optimize it for CoLA expression? And I said, sure, we can do that. And we used the common codons, and sometimes it expressed well, and sometimes it didn't. And it was just a lot of white noise. So we ended up doing the same process for the DOE and the machine learning, but looking at codon optimization. So here the variables are typically for setting 3 instances. For instance, for histidine, you can have CAT or CAG. So we did 50-50 or 25-75 or 75-25, and so on for all the different amino acids. And then you can look at things like RNA thickener structures and runs of Cs and Gs and whatever variables you want to toss in there. And again, this is all published, and you can find the corresponding papers on it here. So we ended up doing 96 sequences for a single chain antibody and for a DNA polymerase. And then on the left-hand side, you see here, this is now measured relative expression yield on the y-axis, and chi. And chi is a measure of how often you use the most common codon. So a chi of 1 means you're always using the most common codon for every single amino acid. And you see there's absolutely no correlation whatsoever. It's just complete garbage, basura all the way. But since these were systematically varied, we can then take that data apart and use that using a prediction of the model space. And so what you're looking at here on the right-hand side is the exact same data that you have on the left, but now plotted as predicted versus measured expression. And what you see is you get a really nice correlation. You get a cross val here of 0.7 or so, 0.6. And then in this case, we also had an additional protein, a fluorescent protein, and a GFP. And then we can again overlay that in the exact same algorithm, and showing that there's a significant correlation between different codon biases in maximizing heterologous protein expression. But it has nothing to do with being the most common codons. There are just certain codons coli like to have in abundance when it's making high production, and some it hates to have. And we can actually capture that in a real live model and using the DOE. So that really changed the way we do recombinant protein expression, and has of course massive effects on biotech as such. And of course, in reality, nobody or academia likes to know about E. coli codon optimization, but of course, in most commercial applications, it comes down to mammalian cell lines. So we did the same thing for, here's an example, and then HEC293. On the left-hand side, you have sort of the DOE design matrix, and each color represents a certain codon bias for different amino acid substitutions. We synthesized these different genes, tested them, and what you see 2 orders, almost 3 orders of magnitude difference between the worst codon optimization and the best codon optimization. And again, there's no correlation whatsoever to common codons, but there are certain codons are just very important, and certain other variables are very important. So we see here in blue is the data set that generated from this first DOE matrix. And off of that sequence, we made a second round that we drilled down further into certain codon biases. And we see another 5-fold or so improvement of expression as a function of recoding the sequences. Again, just focusing on how can we capture sequence function correlation that impacts function of a biological system without any deductive knowledge at all about what is the mechanism, why are certain codons better, why are certain worse. I have no idea, and honestly, I don't really care as long as it works, which I think is the underlying assumption of really all inductive reasoning, and DOE being really a big driver of that. So I'm going to do one more example on protein optimization. So here is something we did with an antibody. So here the customer came to us, we had a starting antibody is something that was a commercial molecule. And for various reasons, we looked at this antibody and we identified 41 positions in the heavy chain that we could introduce substitution based on phylogeny and other information on the variable selection. So heavy chain, there's a total space of 10 to the 12. And light chain, we had 36 positions where we could swap in substitutions, making a space of 10 to the 10. So the total space here is actually, if you look at it, proper math, is 10 to the 23, which of course is so much larger than anything you can ever imagine. And so why do we pick certain amino acids and not others? Well, it's a long story, but really is looking at Mother Nature, looking at all these different IMDb, germline databases, GenBank, epitope prediction, and really looking at what are the substitutions that have been made available in this particular germline, and what is the distribution, how often are they seen, what is the consistency between substitutions in different positions, and so on and so forth. And using that to narrow down this large list of maybe, I don't know, a couple of thousand substitutions that are possible, and rank order them, and then just pick, very naively, just pick the top 50 or so. So then we set up the machine learning, and this is sort of a very simplistic DOE. This is just sort of the top left corner of this big matrix, but the matrix really is a 96 rows. So each row is one amino acid or one antibody. And then in this case, I think we're looking at say 70 or so columns. So every column is one substitution. So for instance, in the top left, we're looking at H13 to G, and the second substitution is L19 to A, and so on and so forth. And every time there's a 1, that means we're introducing that substitution. Right? So it's a fairly sparse data set, but it's, again, going back to the discussion about qualitative space. This is all, it's just ones and zeros. There's nothing else. That's just very, very, very simplistic. Right? I mean, Markus is probably laughing on the low level of complexity of this, but we're just, because everything is qualitative, there are only these two options available. And then we built this nice system that we do for a lot of our fee-for-service business, where we make 96-well antibody proteins. So we can do that nicely in just transient expression and HEC. And just typical example, you do a 96-well plate, and then you look at titer of all these different antibodies, and you get a certain distribution, and fairly nice and even. You can then take that same plate, and you can look at things like binding, for instance. So we're looking at the KD, or 1 over KD, as a function of plate location. So taking that information now, we can build machine learning models where we look at, okay, what is the binding for each of these antibodies that we built, and then we can hide. Typically, we hide, say, 15% from the data. We make a model on the 85%, then we project back in the 15, and then that it'll be a function of the cross val of that particular every single antibody as a function of its location. And of course, so we typically plot that as pink being the model fit, that's with all the data, and cross val being where we hide 15%. And then we do that typically 96 per round, and 96 only because, well, we all know, for whatever reason, I think someone told me Eppendorf back in the 80s set up a plate for some bio company in South City where they did 96, and that just became the standard for everything. But it allows us then to really explore. In this case, we have 60 substitutions along the antibody, we have two options, either the wild type or the substitution. So that space is very, very large. It's 10 to the 18. So as you build this model and you get this correlation, now you can look at the other side of that matrix and pull out the impact for all these different substitutions. So here we're looking at CDR1 in pink, 2 in green, and 3 in blue. And these are now substitutions that affect, in this particular case, binding. So what you see, for instance, is variable 46 is really good. So we have 0 here, that means it's neutral. Everything that's on the 0, that does not affect binding. If it's a positive, it has a positive impact. If it's negative, it's bad for the binding. And then we can then use that machine learning that I've talked about in the previous slide to identify the impact of every single substitution. And what you see here, for instance, so 46 is a great substitution. It's by far the best one in this particular case. And you see the error bar, so that's the indirect effective of the epistatic effects of that, of that substitution. So you see it's fairly narrow, meaning it's not really affecting the distribution as much. And I forgot to say, when we do these tests of 90, when we do this, we hide 15%. We do that by bootstrap. So of course we can do that, 1,000 times and just shake the data in between and hide different 15% and reiterate that process. So that gives us then a measure of what is the relative impact of that particular substitution in all these different, say, 1,000 different contexts. And that will then generate that, let's call it error bar. So for instance, variable 17, not only is it bad, linearly bad, but also is fairly epistatically affected. So it really makes it really hard to predict how it's going to fit in the data set. And then you can use this information, and we typically use this to keep the good substitutions, get rid of the bad substitutions, and test everything that is we don't have enough information about. We can test that in iterative cycles. Sorry. So here's the way we think about these variable matrices, and bear with me, it's a little bit complicated slide first time you see it. But on the x-axis, you have here the antibodies. So in this case, we made 96 antibodies. So each column is one antibody. So the very first antibody has, as you see here, 3 substitutions. This 1, 2, and 3 substitutions. And then each substitution then seen, this particular substitution is seen in antibody number 1, number 3, number, I don't know, 10, 12, and so on and so forth. So that, and in the first set of 96, the blue guys, all the substitutions we're just testing the heavy chain by itself, all wild type in the light, and then the second half as all wild type in the heavy and just substitutions in the light chain. And then we can then take this information. I'm going to skip the yellow round for time constraints, but it doesn't really matter. So then we take this information, and then we see each bad substitution. We can just toss those out. So here, for instance, all these guys here are all bad. We'll just toss them out. We're never going to use them again. This substitution turns out to be fantastic. So we'll just freeze this one in and keep this in this pink round. So the cool thing here, like in the first blue round, the total space we're looking at is 10 to the 23. So that, of course, is a ginormous space. I think 10 to the 23 is, I think, the definition of gazillion. But of course, in this gazillion space, most is garbage. So by just weeding out the bad substitutions, we can very rapidly narrow this down. And that's just by only making 96 sequences in this 10 to the 23 space. We can very rapidly narrow down all this garbage and keep all the good substitutions, and drill us down to, in this case, 10 to the 4, which is all of a sudden a very manageable space. And search that space, and in this case, it's just 81 sequences. So in this experiment, we only did a total of 184 variants for a total space of 10 to the 23, which I think it really speaks volumes to the power of DOE in the sense that how the whole point of the DOE is how can you capture as much information as possible from as few data points as possible, and be able to organize your space search accordingly. So looking at, instead of just looking at the substitutions as they came in, let's look at the activity here for the binding. So here We're looking at the reds, at the blue round, the 10 to the 23 round. So looking at binding now for each of the different variants. So 1/kd. So here's the starting point, and this is the commercial molecule. Had some binding to its target, and then round one, most of them are worse. So a couple of you guys are better, but you have a nice even distribution, which allows you then to really build good models that can correlate nicely with impact on binding. And in the pink round, when we cleaned out a lot of the garbage, not only do we see that we have better hits, but you see the entire population has moved from sort of the blue space in this 10 to 23 space into a much more potent space that, and much more narrow, much more, will funnel this very large space into 10 to the 3. And then we just picked a couple of guys, these last 5 guys, to really drill down binding. And that only captures binding, but in reality, of course, biology is multidimensional. All the fitness you're trying to do is on the very minimal. You always want to make sure you have a good titer of your protein of interest. Maybe you want to have it stable at some pH. You want to have it work for different substrates. All these multidimensional optimizations. So here's the example where looking at this very large space, 10 to the 23. And looking at this, where we did 186 different sequences. So here's the parent, y-axis is titer, x-axis is binding, and then color is temperature. So what we want to do is we want to find something in the top right corner that is very red, right? And then we end up in this case with 4 different solutions, or 3 different solutions plus parent, and you have here on the right-hand side the actual numbers. But that gives you sort of a Pareto front on how you navigate against those 3 different parameters simultaneously. And finally, of course, this is that was just an antibody. Here's a beautiful example we did with Christian Hansen, and here the credit really goes to Christian Jackel, who was the lead scientist on Christian Hansen's side. But we did the same thing now with the enzyme that makes cheese from milk. So how do you coagulate the milk into cheese? And so we engineered this enzyme starting with the chymosin, and that's what you have here on the right hand at the bottom here, the wild type starting point. We wanted to optimize this for expression. So the bigger the circle, the better the expression. When it's really a protease and it cleaves the milk protein, and depending on how it cleaves, you have you may have some unwanted peptide that gives a bitter taste. So the amount of bitter peptide is a function of green versus red. So we want to have it as green as possible, and then we want it to be have as efficient clotting as possible, and which is on the x-axis. And then we're looking at the cutting itself as proteolysis on the y-axis. So here's actually a 5-dimensional optimization if we're making the enzyme for cheese. So in here we looked at 300 variants. The total sequence space is 10 to the 25, and we were actually able, by making less than 300 variants, we can explore this massively large space, and ended up with enzymes that Christian Hansen then physically made those enzymes and actually made cheeses off of them and tasted them, and that at the end of the day is the real assay. And today this is called Chymosin Supreme, and it's the brand coming out of Christian Hansen. So with that, I think that I'd love to have a discussion with Markus and you guys and see what makes sense and how we can see how we can infiltrate the world of biology with a more DOE-based thinking. So with that, thank you very much.
Thank you very much, Claes.